In this video, we will discuss about the third major signaling that is activated when during UPR response. In the start before beginning before start of this before starting this lecture, I would like to correct myself that in the brief introduction about UPR, I have I have written this wrongly as ATF4 in that video. However, uh, the correct gene is ATF6. Correct signal that is activated is ATF6. So what is happening in this? This is the ER membrane. This is the ER membrane, and this part is the ER lumen, that is within the endoplasmic reticulum, and this part is outside lumen, that is cytosol. So usually in the normal condition, what happens is that ATF6 is present at the ER membrane. However, when the stress is observed, this ATF6 is transported by COP2. COP2 is the membrane transport vesicle. COP2 is the protein transport vesicle. Sorry, COP2 is the protein transport vesicle that helps in transport of proteins from endoplasmic reticulum to the Golgi for further modification, post-translation modification. So in this COP2 is also used in case of ER stress to transfer ATF6 to the Golgi, where it is processed as two site, where it is processed at site 1 by protease and site 2 by protease. So what after processing of this ATF6 at Golgi, what will happen is that it will fragment itself in a way that ATF6F is the cytosolic fragment that is exist in this ATF6. However, it is not yet fragmented in when it is at ER lumen. When it is transported and processed by Golgi at site 1 and site 2 protease, then it will fragment itself into ATF6F. This ATF6F is called the cytosolic fragment of ATF6. So this is the cytosolic fragment of ATF6. This is the cytosolic fragment of ATF6. So what happens is that what happens after that, this fragment will then go into the nucleus and act on the UPR target gene. What are the target gene this ATF6 will act? ATF6F will act towards increasing the component that takes part in the ERAD. ERAD, as we have discussed, as we know from our previous videos, that ERAD is the endoplasmic reticulum associated degradation. So ERAD is what ERAD will do. ERAD will do in case of UPR is to increase the degradation pathway and then transfer the protein to the ubiquitation for ubiquitation process. As in UPR, the number of unfolded proteins are more, so we need to increase the flux of the ERAD in order to reduce the stress. So cell acts in that manner when the ATF6 signal is activated. At the same time, it will also work on it will also have another function, that is ATF6 what that function is. As we know that in the first signal, that is IRE1 alpha, it it will splice the XPB1 unsplice into, this is usually present as unspliced in normal condition. In ER stress X, IRE1 alpha will splice this unspliced mRNA, and it is usually called as XPB1 splice. And what it does is it upregulates the chaperones ERAD. It also upregulates the ERAD, and so and what ATF6 does, ATF6 also upregulate the components of ERAD. At the same time, it also upregulate the XPB1. It also upregulate XPB1. So in this way we can imagine that this ATF6 is correlated with the also correlated with the signaling by IRE1 alpha. So if ATF6 is not get activated, then the impact of IRE1 alpha will not be that high enough, as the XPB1 are not present in the cells at upregulated manner. So this is the reason why this ATF6 is also important. So all this we have discussed about UPR response. Okay, this is the third and the major. This is the one of the third signals that gets activated when the ER stress is observed, and that leads to UPR. That's all for this video. Okay. Thank you.